Hello, my name is Gergely Tóth. I'm the CEO and founder of Cantabio Pharmaceuticals. The title of my talk is the following: Development of Pharmacological Chaperones Targeting the Intrinsically Disordered State of Tau or alpha-synuclein as an Anti-Aggregation Therapeutic Approach for the Treatment of Alzheimer's and Parkinson's Disease. As an introduction, let's look at the difference between an intrinsically disordered protein, or IDP, as I will refer to it in this lecture, and a protein with stable tertiary structure. Now, IDPs consist of a dynamic conformational samples which exchange between distinct heterogeneous conformations. I'm illustrating that on the top left, where I'm showing four different structures of as a conformation sample of an intrinsically disordered protein. Conversely, on the right, I'm showing a protein structure that's folded with stable tertiary structure. Now, we estimate that intrinsically disordered proteins make up approximately 40% of the protein coding human genome. IDPs are commonly involved in cell function regulation and signaling, interacting with a wide range of biomolecules. Therefore, IDPs can become very important drug targets. However, the therapeutic targeting of IDPs with small molecules have not been possible so far. Interactions involving IDPs with other folded proteins can be highly specific while be low affinity. IDPs can be hubs in protein-protein interaction networks interacting with multiple protein partners. For example, one IDP can interact with many other folded proteins, or many folded proteins interact with one IDP. In the following, let's look at the structural nature of IDP folded protein interactions. The first case is referred to as coupled folding to binding or disordered to ordered transition. A nice example of this is the p53 MDM2 interaction. In this interaction, the N-terminal of p53, which is disordered, interacts with the first 100 amino acids of MDM2, which is ordered. The resulting complex consists of p53 disordered region becoming ordered, as you can see on the right on the figure in red, forming a fairly stable helical structure. Now, it is likely that the disorder's part of p53 contains what we call a molecular recognition feature. It's really a polypeptide segment in the ITP that is mostly disordered in the free state, but fold during the interaction with a very specific partner. This process is very similar actually to the kind of the induced fit concept that has been around for a long time. So molecular recognition features can mediate binding events in IDPs and be related to the disorder to order transition based recognition. The second case of IDP tertiary folded protein interaction is when the ITP stays disordered during complex formation. IDPs can form disordered or so-called fuzzy complex with ordered proteins, in which the IDP remains disordered similarly as it was in the free state. A nice example of a disordered or a fuzzy complex is the case of the MD15 GCN4 interaction. A simple diagram or a figure of this is shown on this slide, in which in gray is GCN4, which is a stable protein, and MDN15, which is a disordered protein. When they are forming complex, MD15 stays in a disordered state, essentially in a conformational ensemble as it's shown in this figure. Now, this interaction very nicely shows that individually weak and low specificity interactions arising from the essential disordered nature of MED15 can combine to produce a biologically critical function without requiring highly ordered structure. Let's also look at the thermodynamic basis of IDP ordered protein interactions. Disordered binding happens without an unfavorable entropic cost, which is not the case for disordered to ordered transition. In fact, during disordered to ordered binding of an IDP, the loss of entropy must be offset with entopic gains due to specific molecular interactions, whereas in disordered binding, these molecular interactions can be weaker. IDPs can misfold and aggregate, causing the onset and progression of protein misfolding diseases. The aggregation of IDPs is illustrated on the slide. The IDPs have native conformation ensemble that somehow misfolds and forms oligomers, and these oligomers end up transitioning into stable amyloid fibrils. Now we can consider the oligomers from a structural point of view as transient complexes in which the IDP is in disorder. So it's really, so this is basically a disorder in binding complex. Now interestingly, this disorder complex transitions into a stable tertiary folded amyloid fiber, which now we can, from a structural point of view, can consider as a disorder to order transition. Aggregation of IDPs are implicated in the onset and progression of neurodegenerative diseases. This table on the slide summarizes the various IDPs implicated in different diseases. For example, A-beta peptide is implicated in Alzheimer's disease, tau in Alzheimer, Parkinson's, and a number of tauopathies, alpha-synuclein in Parkinson's, and a number of synucleinopathies, TDP-43, ALS, FTD, Huntington in Huntington's disease, prion in mad cow disease. In most of these diseases, these IDPs aggregate and end up gaining a toxic function, and also because they lose their native structural state, they end up having a loss of native function. In these diseases, IDPs end up interacting with their own self to produce specific solid fibrillar pathologies. As the aggregation of IDPs cause the onset and progression of neurodegenerative diseases, disease-modifying therapeutic strategies has been focusing on stopping essentially this aggregation. Now, most efforts have focused on reducing amyloid fiber formation. One reason for that was, or has been, is because targeting the amyloid fibrils has been possible, while, for example, targeting oligomers, which are transient in nature, has been much more challenging. Recently, it has become clear that the most toxic species in this aggregation process are the oligomers. Therefore, therapeutic strategies are needed that reduce both oligomer formation and amyloid fiber formation. Therefore, a therapeutic strategy is needed which not only reduces amyloid fiber formation, but also reduces oligomer formation. One approach could be that we have utilized is to target the native conformational ensemble of an IDP with a small molecule. We need to identify small molecules that bind to the native state of IDP and enhance its essentially native state and function, and reduce its ability to misfold and aggregate into oligomers. Now, the therapeutic strategy to target the native state of a protein to reduce its aggregation has been applied previously successfully. A very nice example of that is tafamidis, which is a drug, a small molecule drug, that has been approved by the FDA for the treatment of trans-striatin amyloidosis. Now, trans-striatin is a tetramer in its native state, and in disease it misfolds and aggregates. Tafamidis binds into the interface of the tetramer, as I'm showing you on the left lower corner, and essentially increases the stability of the tetramer, thereby reducing the likelihood that it can essentially dissociate into monomers and aggregate and form amyloid fibrils. Now, a small molecule binding to a monomeric IDP could impact its structural and biological function in various ways. We have just spoken about misfolding. So a small molecule binding to IDP could stabilize the IDP and thus inhibiting oligomer and fibro formation. Now, a small molecule could also influence its affinity to other interacting protein partners, so it could have an effect in its signal transduction function. A small molecule binding to an IDP could affect the IDP's post-translational modification state because it could essentially reduce the effect of post-translational modifications, and it could possibly even have an impact on cellular localization and its actual levels and turnover. So the question is, can a drug like small molecule bind to an IDP specifically with high affinity? Let's look at first what we know. We know that order proteins have well-defined binding sites in which a very specific small molecule can bind. The mechanism that has been described for this binding mechanism is the key and lock mechanism. Now, in the case of intrinsically disordered proteins, there isn't one static binding site. However, what we know is that there are a number of very different conformations in the ensemble of in the structural ensemble of an intrinsically disordered protein. So the question is, how can a small molecule bind to the ensemble, with what affinity, with what specificity, and with what mechanism? So in order to understand whether small molecules can bind to IDPs, our initial goal was to elucidate whether binding sites existed in the conformational ensemble of IDPs. First, we wanted to just understand this from a theoretical point of view. Therefore, we utilized conformational ensembles of proteins. We searched for hotspots, and then based on the existence of hotspots, we identified binding sites. We did this using structure-based modeling techniques. We utilized this strategy first on alpha-synuclein. We used a conformational ensemble of alpha-synuclein derived from NMR measurements. Now, the search algorithm that was looking for hotspots resulted kind of the two extreme results. If we looked for binding sites on a compound conformation, then we found a number of different binding sites on the compound conformation of alpha-synuclein, as illustrated in the figure on the left. However, when we looked for binding sites on an extended conformation, we found almost no binding sites, as shown on the right. This is obviously pretty logical, because in a compact conformation, more binding sites can assemble than in an extended conformation. Now let's look at in more detail a very specific binding site on a conformation of alpha-synuclein. On the left, I'm showing 3 hotspots that have been identified by the search algorithm. These hotspots are in proximity of each other, forming a good binding site. Now this binding site is formed by polypeptide segments that are essentially far in the sequence of alpha-synuclein from each other, as shown on the right. And therefore you can imagine that if you can bind a small molecule to this very specific binding site, then they act, the small molecule can act as a bridge, essentially stabilizing this whole conformation. The second case that we looked at in more detail was the Aβ42 peptide. We generated in this study, using molecular dynamics simulation, a conformation ensemble for the Aβ42 peptide. We took the conformation ensemble and clustered it and found essentially clusters that had many similar structures of Aβ42, although they were generated at different time points in the molecular dynamics trajectory, which suggested that certain conformations refers essentially transition from one conformation to another, but in time they can transition back to this original conformation. Now we took these representatives from each cluster and searched for hotspots and binding sites, and found in fact that different binding sites existed in these clusters. What's really interesting that certain well-populated clusters had well-defined binding sites within them. An example of this is on the top right, where we show binding sites in the middle of the structure of Aβ42 from cluster 2. We could also take these essentially conformations and dock well-known inhibitors of Aβ42 onto them, for example, which we did with curcumin and Congo red on the bottom right. This study showed us that binding sites can exist, small molecule binding sites can exist for Aβ42 peptide. The third protein system that we looked into in more detail was tau. We used two NMR-derived conformation ensemble of tau and looked for binding sites, which we found. Next, we compared the propensity of having a binding site on monomeric tau versus the propensity of having a binding site on a folded ordered protein, as shown in the bottom left diagram. Now we found that the propensities are very similar, interestingly, for monomeric tau to have a binding site versus a folded protein. Next, we used the binding sites that we identified on the monomeric tau ensembles to probe where methylene blue, a tau aggregation inhibitor, could bind to monomeric tau. Now we found that methylene blue may be binding very close to cysteine 291 and cysteine 322, and also essentially elucidated a binding mode and a complex structure between methylene blue and monomeric tau. Further, based on this, we went and tested whether methylene blue bound to monomeric tau, and found that using microscale thermophorezes that in fact monomeric tau bound methylene blue quite with high affinity. Full-length human tau bound methylene blue with a KD of 266 nanomolar, and K18 tau bound methylene blue with 125 nanomolar. Interestingly, similar results were reported by Zhirong Liu's group, who have surveyed a number of intrinsically disordered proteins and reported that, in fact, druggable binding sites may exist on intrinsically disordered proteins. Moreover, they performed molecular dynamic simulations similar to us on Aβ42 and showed that small molecules can bind to flexible proteins, and they used the phrase ligand clouds around protein clouds as a mechanism for small molecule protein binding. Again, resembling the disordered to disorder complex formation that I suggested to happen for protein for IDP interaction, for example, during the aggregation process forming transient oligomers. We decided to apply our theoretical findings on targeting the native state of IDPs in small molecules in Alzheimer's and Parkinson's disease. Now, Alzheimer's and Parkinson's disease, these are chronic degenerative disorders. There is a gradual neuronal death as the disease progresses, which results in loss of memory, cognition, motor function, behavior changes, and eventually death. An example of how when neurons degenerate in Alzheimer's disease is shown on the right, where healthy human brains essentially shrinks, and there's atrophic degeneration of the brain. As I highlighted, very specific proteins aggregate mostly in Alzheimer's or Parkinson's disease. In Alzheimer's, A-beta peptide aggregates, forms plaques. Tau protein aggregates, forms neurofibrillary tangles, and alpha-synuclein aggregates into fibrillar aggregates and localize in Lewy bodies. The first applied study that I'd like to talk about in more detail is when we targeted monomeric alpha-synuclein by small molecules. In this study, we identified binding sites in a conformational ensemble of alpha-synuclein and used these binding sites as a starting points for high throughput docking screen, in which we identified small molecule fragments that bound to these essentially theoretical binding sites. We ended up testing over 30 compounds for their ability to inhibit aggregation and reduce alpha-synuclein toxicity caused neuronal death. And one of these compounds worked very efficiently. This compound is shown on the top right. This compound is ELN 484-228. And this compound was able to alleviate a loss of dopaminergic neurons and neural retraction induced by A53 threonine mutant of alpha-synuclein in primary rat embryonic midbrain cultures. Essentially, in this experiment, we overexpressed alpha-synuclein A53 via viral vector in rat primary neural mixed cultures, and found that the TH plus neuronal viability was reduced. And when we pre-treated with ELN 484-228, we saw a significant alleviation of neuronal loss and neural length reduction, suggesting that this compound is able to protect from alpha-synuclein toxicity caused aggregate toxicity. After applying computer-aided drug discovery methodologies to target monomeric IDPs, we decided to pursue experimental screening methodologies. We identified a very unique methodology and applied it on tau. In this study, we used high-throughput chemical microarray surface plasma resonance screen to identify small molecule binders to monomeric tau. We did this the following way, which is summarized on the figure on this slide. We used a chemical microarray where small molecules are immobilized on a linker. Then we incubated and added tau to this essentially microarray and detected a binding between the monomeric tau and the immobilized small molecule with surface plasma resonance. We partnered with this study Biography Pharmaceuticals, now Novalix, who developed this chemical microarray screening technology. We ended up screening over 110,000 small molecules and ended up getting moderate signal-to-noise ratio with a hit rate of about 0.76%. After chemoforensics analysis of the hits, we identified 11 novel scaffolds and 42 singletons over about 118 compounds that were identified to bind to tau. On the basis of this, 70 compounds were selected for synthesis for hit follow-up. We tested the 70 compounds for the ability to reduce the aggregation of tau in vitro. We tested the compounds on 3 different constructs of tau, on full-length tau, on K19 and K18 delta 280. In general, we found that the compounds were able to reduce the aggregation of K18 delta 280 the most, and much less for full-length tau. Although I have to highlight that for K19 and for H-tau40, for full-length tau, we used heparin to induce the aggregation, while we did not use that for K18 delta 280. Now, heparin may be competing for binding to monomeric tau with the small molecules, and so it could be impeding for their full ability to reduce tau aggregation. The diagram shows the extent of the ability of the small molecules to reduce aggregation of K18 delta 280, and essentially what we can see that surprisingly most of the compounds were able to essentially have an effect in reducing tau aggregation, while we also find a few compounds that actually ended up increasing the aggregation propensity of tau. Next, we tested the compounds for the ability to reduce tau aggregation in cells. For this, we used a cell model where we applied N2A cells induced to overexpress tau K18 delta 280. We detected aggregate formation using thioflavin S fluorescence. In general, what we found that most of the compounds had some impact on reducing essentially tau aggregation in the cell model, and there were some compounds or a few compounds that actually reduced aggregation even in higher with higher efficacy than we found in the in vitro model previously. We publicized hits from this screen in a peer-reviewed journal called Current Alzheimer's Research a few years ago, and have in parallel taken forward the most drug-like compounds and developed them further with the aim of developing drug candidates as disease-modifying treatments for Alzheimer's disease. One of the compounds that we have been developing at Cantabio Pharmaceuticals from this study is CB81. Now, CB81 inhibits tau aggregation in vitro and in cells. On the left, we're showing an aggregation assay of tau with and without CB81, and what this illustrates that CB81 is able to completely inhibit the aggregation of tau in vitro. On the left, we're showing again the same N2A cell model where essentially CB81 is able to reduce the formation of thioflavin S positive tau aggregates in a dose-dependent manner. Now, next we tested 582451 or CB81 in a Drosophila model of tauopathy, in which we overexpressed human tau in the motor neurons of Drosophila. This results in a locomotor impairment, which you can quantify, for example, by essentially the climbing speed of Drosophila. Drosophila overexpressing tau in its motor neurons causes a reduction in climbing speed, as you can see in this figure on the bottom. And when we feed the compound 582451 from essentially day 1 to Drosophila, then we are able to alleviate this locomotor impairment, or essentially the climbing speed reduction, as shown in this diagram. And this actually, this reduction, this elevation is very, very significant. We also apply the same screening strategy to identify small molecule binders to monomeric alpha-synuclein. In this study, we used monomeric alpha-synuclein to screen using high throughput chemical microarray surface plasmon resonance screen, and essentially identified also a plethora of hits. The screen resulted in a hit rate of 0.49%. Now, interestingly, in the alpha-synuclein screen, we identified a large number of positively charged compounds, and probably the reasons for that is that the C-terminal of synuclein is negatively charged. Conversely, in the tau screen, we identified a large number of negatively charged compounds, and probably the reason for that is because tau is a positively charged protein. This also suggests that our screen identified specific compounds to these two different intrinsically sorted proteins. Now, we took forward also a number of compounds from this screen and tested them in alpha-synuclein aggregation assays. From among the hits that we identified, we published 3 compounds that I'll talk about today. These 3 compounds' ability to reduce alpha-synuclein aggregation is shown on this slide. On the top 3 diagrams are 3 distinct compounds. From among them, 576-755 was the most potent or effective. In fact, this compound was able to reduce non-seeded fibrillation and also seeded fibrillation of alpha-synuclein, and was able to importantly reduce alpha-synuclein aggregation in a dose-dependent manner, and showed near half maximal anti-fibrillation activity at sub-stoichiometric concentration of 25 micromolar in this fibrillation assay when alpha-synuclein was incubated at 70 micromolar. Next, we tested the 3 hit compounds in an alpha-synuclein oligomerization assay. We used 2 different types of oligomerization assay. The first one on the top left is the diagram shows the results from a oligomerization assay in which we used the split Gaussian luciferase complementation system to detect alpha-synuclein aggregation, and what we found that 576-755 was the most potent with an IC50 of about 10 micromolar. We also use a FRET-based oligomerization assay shown on the right. And in this oligomerization assay, all 3 compounds had an impact in reducing the oligomerization of alpha-synuclein, and again, 576-755 was the most potent. Next, we used and tested 576-755 in an oligomerization assay in cells. Again, this was a split Gaussian luciferase assay, and what we found that the compound was able to reduce oligomerization of alpha-synuclein. Now, at the same time, 576-755 did not significantly change the levels of alpha-synuclein shown by Western blot. Next, we tested whether 576-755 is able to alleviate the loss of downverging neurons due to alpha-synuclein overexpression. For this, we used a rat primary neuronal culture, mixed culture, where we overexpressed alpha-synuclein A53.3. This overexpression causes a loss of TH+ neurons, as shown in the diagram. On the right, when we pre-treat with 576, 755, we find and observe that we alleviate TH plus neuronal loss, suggesting that we are protecting against dopaminergic neuronal loss in this cellular system. As I arrive to the end of my lecture, I'd like to summarize. We find that the native monomeric ensemble of IDPs, such as alpha-synuclein, tau, and A-beta, contain transient small molecule binding sites. Furthermore, we find that diverse novel drug-like small molecules can bind to the monomeric soluble tau and alpha-synuclein. We identified dozens of novel small molecule binders to monomeric tau and alpha-synuclein via biophysics-based screening approach. We find that selected novel small molecules bind to monomeric full-length tau or alpha-synuclein, also inhibit the protein's aggregation in vitro and in cells robustly. Selected small molecules that bind to monomeric full-length tau were able to rescue tau overexpression caused phenotypes in Drosophila. Moreover, selected small molecules that bind to monomeric state of alpha-synuclein was able to alleviate dopaminergic neuronal death due to alpha-synuclein overexpression. In conclusion, targeting the monomeric form of tau and alpha-synuclein by small molecules may be a feasible drug discovery strategy for the treatment of Alzheimer's and Parkinson's disease. Thank you for your attention. I'd like to emphasize that this research was done in partnership with the following contributors: Professor Eckhard Mandakov and Ivan Mandakov and colleagues at the Structural Biology Unit of Max Planck Institute, Professor Christopher Dobson, Professor Peter St. George Hislop, Professor Franklin Albireo, and Dr. Zhao, University of Cambridge, a number of people from Gergely Pharmaceuticals, including Thomas Newman, people from Elan Pharmaceuticals, Dale Schenk, and especially would like to thank Lisa McConnell, who's now a professor at UCSF, and colleagues at Cantabio Pharmaceuticals.